Hello student, today the topic which I am going to discuss is Golgi body or Dalton complexes or lipochondria. Now this cell organelle was first observed by George, okay, and later the another scientist known as Camillo Golgi, okay, he gave the morphological structure of Golgi body or Dalton complexes was given by Camillo Golgi. Okay, in the nerve cells of cat and bird own. Okay, so this morphological structure of Golgi body in the nerve cells of cat and the owl was given by Camillo Golgi. Another scientist who came up and he had given the ultrastructure of the Golgi body. Now the term Golgi body, it is also known as dictosomes. Okay, dictyosomes. Now, this term were specifically used in plant cells. The term dictosomes are used only for plant cells, but dictosomes is also known as Golgi body only. But for animal cells, we use Golgi body, Dalton complexes, or lipochondria. Now, if we see, the Golgi body exists in two forms. One is in the localized form, okay, and one is in the diffuse form. Now, dictosomes are in the diffuse form, okay. Diffuse form means scattered, okay. Diffuse form means scattered. Scattered in the cytoplasm. Okay. Now, scattered in the cytoplasm. So those Golgi body, which in the diffuse form, they will be scattered in the cytoplasm and they are not easily distinguishable. Whereas in localized form, they will be near to the nucleus. Okay, those examples like in vertebrates, okay, and animal cells. You will find in the case of vertebrate cells and animal cells. You will find that the Golgi body is in the localized form, that is near to the nucleus. Now, the shape of the Golgi body, it is said to be pleomorphic organelle. It means that it is pleomorphic. When we say pleomorphic, it means Golgi body, they changes their shape, okay, depending on the functions. According to the functions, they change their shape. According to their functions, Golgi body changes its shape. Okay, that is said to be pleomorphic. Now, the number of Golgi body, there will be variation in the number of Golgi body. In the case of, in every animal cells, you will find at least one Golgi body. Okay. Whereas in the case of plant cells, you will find around 6 to 12 number of Golgi body in the case of plants. Now, depending on the numbers of, depending on the size, if you see the size of the Golgi body, some of the Golgi body are smaller in size and some are large in size. Large. small size. Just write as Golgi body, G. Now, this large size means those cells which are secretory cells, okay? Secretory cells. Secretory cells means those secret, okay? And these are non-secretory cells. So you can take an example of liver cells as a secretory cells, and non-secretory cells like muscle cells. You can also take an example like pancreatic or liver cells, which are secretory cells. Okay, so you will find that in the secretory cells, large Golgi body, okay, are found, whereas in non-secretory cells, example like muscle cells, small Golgi bodies are observed. Okay, so this is the basic information about the Golgi body. Now we are going to discuss in a brief the structure of the Golgi body. Now this structure was explained, okay, was given, the electron microscopic structure was given by Dalton. Electron microscopic structure was given by Dalton. And basically it has three elements. The first element is cistern. Then we have vesicles, and then we have vacuoles. These vesicles are tiny and these are large. So these are the three basic components, and this Golgi body is said to be polarity. I'll explain to you what does polarity means. Polarity means in one of the side it will be concave and the another side is said to be convex. And if it is concave side, it means that it is toward the plasma membrane, and if it is convex means toward the nuclear membrane. Toward the nuclear membrane. Okay. Now I'll draw the structure and then I'll explain to you. Three elements, cisternae. Okay. So you can see that it is flattened sac. So these numbers of these are called to be cisternae. In the case of animal cells or plant cells, you will find many cisternae. Okay, these are said to be cisternae. And this has a connected, one cisternae is connected with another cisternae like this. Okay. Now these are, in the case of plants and animals, many cisternae have been observed. Whereas in case of fungi, okay, it is unicisternae. Means the Golgi body has only one cisternae. Unicisternae in fungi. Okay. Means Golgi body. Okay, that is unicisternae. Means only one cisternae in case of fungi. This is very important. In the case of plants or animals cells, you will find many cisternae. Now, you will find at the concave. Okay, this side is said to be the concave or the maturation phase or the trans phase. Okay, so here you will find spherical. Okay, and this is said to be spherical shape elements called vacuoles, large vacuoles, and small tiny, okay, so these are said to be vesicles. These toward the concave side we call as maturation or transphase. Maturation or transphase towards the plasma membrane. Okay. Now this side is said to be the convex side, and these are said to be formative phase or cis phase. Okay. Now these constitute your Golgi body. Okay, we have a vacuoles, we have a cisternae. Okay, we have a vesicles. Toward the concave phase we call it as maturation or transphase. Toward the nuclear membrane it is said to be formative phase or the cis phase. As I said that if this is a nucleus, if this is a cell, there is a nucleus, right? And from the nucleus there is a extension, and that extension is nothing but endoplasmic reticulum. So your Golgi body, okay, your Golgi body is near, this formative phase is near to the endoplasmic reticulum. You have to remember this point, okay? The formative phase or the cis phase is near to the endoplasmic reticulum. With this structure, let's try to understand the functions of Golgi body. First important function is secretion. We're talking about the functions of Golgi body. Secretions and packaging. For example, like polysaccharide, okay, polysaccharide or halogen. These are secreted by the Golgi body. Example like secretion of hormones. Hormones are also secreted by Golgi body, like thyroxine. The third functions can be, it is said to be yolk formation. Okay, that is VITTELLO genesis. Okay, vitellogenesis is nothing but yolk formation. Another functions can be cell plate formation. Cell plate formation. Secretions of your hormones, secretions of your polysaccharide, halogen, okay. Vitellogenesis, that helps, means yolk formation. They help in yolk formation, in the cell plate formation, and also in the synthesis or in the secretion of the middle lamella, okay, which is a pectin. Now the most important point, the last functions that I want to discuss is glycosylation. Glycosylation. Now what is glycosylation? If carbohydrate is added, okay, to a protein, okay, if carbohydrate is added to a protein, we call glycoprotein. Glycoprotein. Now, this, when you add carbohydrate and protein, it is a oligosaccharide. Oligosaccharide. Now, this glycoprotein helps, okay, it helps in identifying, okay, in identifying the cell, okay. I'll write as identifying the cell or cell identification. For example, one person have to be, there is a organ transplantation. Then, when an organ transplantation takes place, two persons will have different glycoprotein. And this glycoprotein should be matched during organ transplantation. Okay. There are many functions of this Golgi body, but these are some of the important functions that I have explained to you. Now in the next class, we will discuss about ribosome. Thank you.